an enzyme variant created by engineers and scientists at the University of Texas at Austin, can break down environment-throttling plastics that typically take centuries to degrade in just a matter of hours to days. This discovery, published in the journal Nature, could help solve one of the world's most pressing environmental problems, that is, what to do with the billions of tons of plastic waste piling up in landfills and polluting our natural lands and water. The enzyme has the potential to supercharge recycling on a large scale that would allow major industries to reduce their environmental impact by recovering and reusing plastics at the molecular level. The project focuses on polyethylene terephthalate (PET), a significant polymer found in most consumer packaging, including cookie containers, soda bottles, fruit and salad packaging, and certain fibers and textiles. It makes up 12% of all global waste. The enzyme was able to complete a circular process of breaking down the plastic into smaller parts, that is depolymerization, and then chemically putting it back together, that is repolymerization. In some cases, these plastics can be fully broken down to monomers in as little as 24 hours. Researchers at the Cockrell School of Engineering and College of Natural Sciences used a machine learning model to generate novel mutations to a natural enzyme called PETase that allows bacteria to degrade PET plastics. The model predicts which mutations in these enzymes would accomplish the goal of quickly depolymerizing post-consumer waste plastic at low temperatures. Through this process, which included studying 51 different post-consumer plastic containers, 5 different polyester fibers and fabrics, and water bottles all made from PET, the researchers proved the effectiveness of the enzyme, which they are calling fast PETase, that is, functional, active, stable, and tolerant PETase. The researcher says that this work really demonstrates the power of bringing together different disciplines, from synthetic biology to chemical engineering to artificial intelligence. Recycling is the most obvious way to cut down on plastic waste, but globally, less than 10% of all plastic has been recycled. The most common method for disposing of plastic, besides throwing it in a landfill, is to burn it, which is costly, energy intensive, and spews toxic gas into the air. Other alternative industrial processes include very energy intensive processes of glycolysis, pyrolysis, and methanolysis. Biological solutions take much less energy. Research on enzymes for plastic recycling has advanced during the past 15 years. However, until now, no one had been able to figure out how to make enzymes that could operate efficiently at low temperatures to make them both portable and affordable at large industrial scale. Fast PETase can perform the process at less than 50 degrees Celsius. Up next, the team plans to work on scaling up enzyme production to prepare for industrial and environmental application. The researchers have filed a patent application for the technology and are eyeing several different uses. Cleaning up landfills and greening high waste producing industries are the most obvious. But another key potential use is environmental remediation. The team is looking at a number of ways to get the enzymes out into the field to clean up polluted sites.